Hello, hi, a very happy good evening. So this is Krishna and we've been talking about the story of life on earth, and that's biology. So welcome to my new beginning series. Hi Anures, hello, very happy good evening. So thank you for joining my lecture today. So thank you for joining on a weekend. So that's really great. So I hope you guys are doing well, and please don't forget to like, share and subscribe. So today we're going to talk about the most interesting part. So we are going to talk about the structure of the human heart. So all of us know it's four-chambered, but we'll get the structure properly cleared. So which side, what are the valves that is present, so what is the notch, so we will get everything cleared so that it's easy for you. So today we'll talk about the structure of the human heart, and on Monday we will see how this heart functions actually. So on Monday we're going to talk about the cardiac cycle. So today we are going to talk about the structure of the human heart. So with that introduction, so let's move into today's lecture. So in this chapter, so we have been talking about blood and its components. Then we went on to talk about what is blood grouping. So we spoke about ABO blood grouping, we spoke about RH antigen. So we also added on to what is erythroblastolysis fetalis. So today we'll have a quick recap, and then we'll talk about the structure of the human heart. So before we talk about the structure of the human heart, let's also talk about what is lymph, that is a tissue fluid, and what is open and closed circulatory system, and then we move into the structure of the human heart. So structure of the human heart is really very simple, so I can actually finish it off in 5-6 minutes. So I thought I will finish the remaining portions, that is lymph, what is open and closed circulatory system. So we'll finish up all this and then move into the structure of the heart, right? So let's move forward. So blood grouping, so I guess you guys remember blood grouping. So we were doing blood grouping yesterday in class. So for all those who attended my lecture, so you would have understood, and others, I believe you have watched the recorded version of it. So now let's talk about blood grouping. So there are two types of blood grouping. One is ABO blood grouping and the other one is RH antigen. So ABO blood grouping, so it is based on the blood group A, B, O and AB. Right? So on what basis your blood group is decided? Your blood group is decided based on the antigen that is present on the surface of your RBC. So let's talk about ABO blood grouping first. So ABO blood grouping, I told you, is controlled by gene I. So genes have an alternate forms of itself, and that is known as alleles. So we also spoke that it has 3 alleles, A, B and I. Right? Okay. So now moving further. So depending on the antigen that you have on the surface of your RBC, your blood group is decided. So based on the antigen that is present on the RBC, the blood group is decided, and we have the corresponding antibody which is present in the plasma. So by now you should know what is the difference between blood and plasma. So blood has two components. So let's revise that as well. So components of blood are divided into formed elements and the other half is plasma, that is the fluid portion of the blood, right? So the formed elements include your RBC, the WBC and the platelets, right? So your plasma is the liquid portion, so it has 90% of water, you can say 90 to 91% water. So remaining, it has what is known as the plasma proteins. So we have 3 plasma proteins, that is globulins, albumins and the fibrinogens. So I guess this base is clear. So now we went on to talk about that because I said plasma contains antibodies. So Suhani Mathur, yes, hello, okay, so you're saying good night, okay, so that's good evening, a very happy good evening. Thank you for joining my lecture today, so please do like, share and subscribe. So let's move forward. So suppose when I say A antigen is present on the surface of RBC, the corresponding blood group is A, and the antibody that is present in your plasma is B. So when I say the blood group is B, when I say the antigen is B, the blood group is B and the antibody is A on the plasma. So when I say the antigen is A and B, so the blood group is AB group, and the antibody, there is no antibody for the blood group A and B. So moving forward, talking about when there is no antigen, the blood group is O, but it has 2 antibodies. So this is your ABO blood grouping. So your O blood group is the universal donor. Then you have AB blood group, which is the universal recipient. Fine. So now moving forward, we went on to talk about what is RH antigen. So apart from the antigen AB, so there were also another antigen that is known as the RH antigen. So why is it called as the RH antigen? Because it was initially found in the rhesus monkey. So that is the reason we call it as the RH antigen. So when you have the RH antigen on the RBC, so it is considered positive. So when you do not have this RH antigen, it is called as negative. So based on this case, we spoke about a condition that is erythroblastolysis fetalis. That is when the mother is RH negative and the fetus is RH positive. So at the time of pregnancy, the mother and the fetus are separated by placenta. But at the time of child delivery or the delivery of the baby, that is parturition. So what happens? There are high chances that the mother's blood and the baby's blood gets mixed, right? So when the mother's blood comes in contact with the baby's blood, the mother's blood does not have this antigen. So according to the mother, this is foreign. So the mother's body starts producing antibody, but by the time the mother's blood starts producing antibody, the baby is delivered outside, so the baby is pretty safe. Okay, so now what happens when the second baby is also in the same condition, when the mother conceives for the second time, the antibody that is produced in the mother with respect to this antigen is stored as a memory. So for the second baby, even before the delivery can happen, the mother's blood will produce antibodies and kill the RBC of the blood, thus causing severe anemia. So that is erythroblastolysis fetalis. So we spoke about this condition yesterday, right? So this is what we were discussing in yesterday's lecture. So now moving forward, yeah, we spoke about ABO blood grouping and RH antigen. Yes, so we are done discussing both of them. So now what is tissue fluid? So, so before we go to tissue fluid, so we have to talk about these two terms. So what is it? So you all agree tissues are made up of group of cells. So we have group of cells which makes up the tissue. Yes, Bhavya, a very happy good evening. So thank you for joining my session. That's completely fine. So please do like, share and subscribe. So I was just revising yesterday's portion. So I can see you're on the right time. Okay, so today we want to talk about what is tissue fluid. So tissues are made up of groups of cells, right? A tissue is vascular, which means it is connected with blood vessels. Sometimes water-soluble substances and other certain molecules escape from the blood vessel and they leak into the spaces between the tissues. So ideally a tissue is supplied with blood vessels. Sometimes certain molecules, they escape or leak from the blood vessel and move into the spaces between the tissues like this. So now since this is tissue and this fluid is present in the cells between the cells in the tissue, it is known as tissue fluid. Now someone has to take up this fluid and put it back into circulation, right? So that is how it is. So now who carries this is a network of vessels, that is your lymphatic vessels. The lymphatic vessels carries these fluid that has leaked between the cells in the tissue and puts it back in the circulation. So that network of vessels is known as lymphatic vessels, and the fluid that flows inside the lymphatic vessel is the lymph. So lymph is a colorless fluid that also plays a role in the defense mechanism of the body. So if you can see, the green color network is the network of the lymphatic system, okay? So once again, Angle Thakur, yes, good evening. So thank you for joining my session today. So please do like, share and subscribe. So once again, so cells comprise of tissues, right? So your tissues are made up of group of cells, to put it in another way. So blood vessels are supplied to the tissues. Sometimes certain molecules from the blood vessels, they leak and come inside the spaces between the cells. So now this is a tissue, so your fluid is here, so it is called as tissue fluid. Now someone has to collect the fluid and put it back into circulation. So who would do that? So you have a network of vessels known as the lymphatic system. So the lymphatic system takes up the fluid and puts it back into circulation. So this fluid, once inside the lymphatic system, it is called as the lymph. So lymph is a colorless fluid that plays an important role in the defense mechanism of the body. Okay, fine. So this is about lymph. I guess lymph is clear. So now we're going to talk about what is open and closed circulatory system. So from the term, I guess it's clear, or if you have read animal system, so these two terms should be clear. What is open and closed circulatory system? So let's talk about open circulatory system. So ma'am, open circulatory system means there is no heart, there is heart, there is blood vessels. But then what is the difference? So let me say, in our body, so we are talking about higher vertebrates, that is, for example, humans, let's say. So you have the heart, you have the lungs, you have the liver, you have the kidney, right? So heart pumps in oxygenated blood, right? So from the heart, it separately goes to the liver, it separately goes to the lungs, and separately goes to the kidney. So this is through hepatic artery. So this is through pulmonary vein. So sorry, this is through again, okay, so from the heart, actually from the lungs it goes to the heart. So again, this is the renal artery, right? To each organ it moves separately. So here the blood vessels are well developed. So that is closed circulatory system. So in open circulatory system, so I have a depression like this, I have an opening like this, then I have the heart here. So the heart pumps in oxygenated blood into this cavity and all the organs are bathed or dipped inside this cavity. Then the deoxygenated blood goes to the heart. The heart is not connected to each and every individual organ unlike the higher vertebrates. It is just dipped inside a cavity and the blood vessel is supplied into that cavity. So this is closed circulatory system. So closed circulatory system are seen in your arthropods and in your molluscas. So arthropod is the phyla which consists of insects, and then we have molluscas. So molluscas are shelled organisms. So your closed circulatory system is seen in all chordates, that is vertebrates, you can say. So chordates include your cephalochordata and your urochordata as well. So all the chordates and annelida, that is the earthworm-like creatures, they have closed circulatory system. So among these two, which is more advantageous or beneficial is the closed circulatory system because the flow can be regulated. So I can regulate the flow of blood that is moving into the kidney, right? So here I cannot regulate because it is common for all the organs, right? So this is more advantageous compared to the open circulatory system. So this is about open and closed circulatory system. Ma'am, the Telegram link in the description box is for JEE, not NEET. Okay, so I'll ask the technical team to get it changed. Okay, Bhavya, I'll ask them to do. Thank you for bringing it to my knowledge. Fine. So I guess open and closed circulatory system is fine. So here I have a definition, a proper difference between the open and closed circulatory system. So first is open. In this system, the blood is pumped by the heart through large blood vessels into the body cavities called sinuses. So I said depressions, so sinuses are also depressions, so that is what they said. So in this system, blood is pumped by the heart through a closed network of blood vessels. So that is the primary difference. In both the cases, you have blood vessels, but in the closed circulatory system, it is well developed, okay? So the body tissues are in direct contact with the blood. The body tissues are not in direct contact with the blood because it is, so body tissues are not in direct contact with the blood because each one has a separate vein and an artery that is moving inside, right? So the body flows, the blood flows at low pressure here, hence it is slower and a less efficient system. But whereas here, blood flows at a higher pressure, so it is faster and more efficient. The flow of blood is not regulated through the tissues and organs, whereas here it can be regulated by valves. This system is present in arthropods and molluscas, but this is seen in annelids, echinoderms, and your vertebrates. So this is the difference between your open and closed circulatory system. Fine. So talking about the circulatory system, so circulatory system includes your blood, your heart and your blood vessels. So we are done talking about the blood. So today we're supposed to talk about the heart, right? So the heart is mesodermally derived organ. So when I say mesodermal, so it is a type of embryonic germ layer. So at the time of embryonic development, there arises a middle layer that is mesoderm. So it is an embryonic middle layer. So that layer gives rise to the heart. It is situated in the thoracic cavity because at the dorsal side of the heart, you have the thoracic vertebrae. So it is situated in the thoracic cavity in between the two lungs, but slightly tilted towards the left. So this is the location of the heart. Yeah, so this is how the heart is. So it might look complicated, but we're going to simplify the structure of the heart into the common way that we can remember. So we are not going to complicate the structure of the heart like this. Though the heart structure is like this, we will simplify it according to our way. So let's move forward and talk about the core topic for today, that is the structure of the human heart. So we know the human heart is four-chambered. Yeah. So let me draw it here. So human heart is four-chambered, so I am drawing four chambers, right? So we have, what are the four chambers? So talking about the four chambers, so the four chambers are atria. So we have two atria and we have ventricles. So we have two ventricles, right? So these are the four chambers. Now, how it is divided? So this is right atria and this is right ventricle. This is left atria and this is left ventricle. Fine. So now the four chambers are marked. Fine. So the heart is double layered just like your lungs. So the membrane of the lungs is known as pleural membrane. So like that we have a membrane here, that is pericardial membrane. So in between the atria, there is a very, very thin wall. There is a very thin gap. In between the ventricle and the atria also, there is a very, very thin wall. In between the ventricles, I have a very, very thick wall. So this wall is pretty thick. So this is a thicker wall. Fine. So now let's mark the parts so that it is clear. So the heart is double layer, it is double membrane. So this membrane is known as pericardial membrane. Right? So inner pericardial membrane, outer pericardial membrane. So this is outer and this is inner, and the space is filled by pericardial fluid. So this is between the two atria, so it is known as this is intra-atrial septum. This is between the two atria. This is intra-atrial septum. So septum is wall. Okay. So now in between the two, in between the atria and the ventricle, it is called as the atrioventricular septum. Atrioventricular septum. Fine. So in between the two ventricles, it is called as interventricular septum. Fine. So this is pretty thick and the remaining are thin. Okay, so we have valves in the heart to prevent the backflow. Suppose if the kidney is sending deoxygenated blood to the heart, it should not come in reverse direction. That is wrong. So that is a malfunctioning of your body. Okay, so to prevent the backflow, there are valves present in your heart. So there is the valve that is present here, and there is another valve that is present here. There is also a valve here, and there is also a valve here. So what are these valves called as? The valves that is beneath the ventricles, both the valves have the same name. So this is known as semilunar valves. Okay. So in between the left atria and the left ventricle, there is a valve that is known as bicuspid valve or mitral valve. Fine. So in between the right atria and the right ventricle, it is called as the tricuspid valve. Fine. So the parts of the heart are marked. Is it clear now? Yes. So we have 4 chambers: right atria, right ventricle, left atria and left ventricle. Okay. So these 4 chambers are surrounded by double membrane. So the membrane is called as pericardial membrane. So we have an outer pericardial membrane and an inner pericardial membrane. The space between the 2 membrane is known as pericardial fluid. So in between the 2 atria, we have a very thin wall. The septa is wall. Okay. So this is very thin. So this is intra-atrial septum. Fine. So between the 2 ventricles, the wall is very, very thick. So that is interventricular septum. So between the atria and ventricle also there is a wall. There is a wall. Wall is septum. So that is atrioventricular septum. Now, to prevent the backflow, there are valves that is present inside your heart. So, beneath the right ventricle and the left ventricle, you have a valve known as semilunar valve. So, beneath the right atrium and the right ventricle, so you have a valve that is known as tricuspid valve. Between the left atria and the left ventricle, we have the bicuspid or the mitral valve. So, the structure of the heart is clear? Yes? Hi sweetie, hello. So I hope you're doing good. So thank you so much for joining my session today. So please do like, share and subscribe. So this is clear, the structure is clear. So what are the different septa and different septae and the valves that are present in the heart? I guess this is clear. So now let's talk about how and where the blood comes inside. So let me redraw the structure of the heart so that it's clear. So this is the first part. Okay. So I am drawing it again so that we can talk about the next part. So this is right atria, right ventricle. So this is left atria and the left ventricle. Right? I am just redrawing because here I explained you the structure. Now I have to explain the flow. So blood moves inside the heart through the right ventricle. So who brings the blood inside the right ventricle is the superior or the inferior vena cava. So superior vena cava brings deoxygenated blood from the brain and the neck portion and puts it inside the right atrium. So inferior vena cava draws blood from the lower parts of the body and puts it inside the right atria. So once inside the right atria, we have a valve. So this is the tricuspid valve. Then the blood moves, so this valve opens and the blood moves inside the right atria. Okay, so once inside the right atria, so we have a semilunar valve. So now this valve will open and the blood will move inside pulmonary artery. Where it will move? It will move inside pulmonary artery. Fine. Now from the pulmonary artery, it will move inside the lungs. So in lungs, it will mix with the oxygen and it becomes oxygenated. So it is all deoxygenated blood, right? So this is carrying deoxygenated blood. So now it moves inside the lungs to get oxygenated. So now through the pulmonary vein, so now the pulmonary vein is carrying oxygenated blood. Fine. So now this pulmonary vein moves inside the left atria. So again I have a valve. This is the bicuspid valve. And from the right atria, it moves inside the left ventricle. So inside the left ventricle, again there is a semilunar valve which opens and the blood moves inside aorta. So aorta distributes the oxygenated blood to all parts of the body. Is it clear now? So is it clear? Is the flow clear? So this is the bicuspid valve. So this is the semilunar valve. Sorry, this is tricuspid. I'm sorry. So this is the tricuspid valve, the bicuspid valve, and this is semilunar valve. So blood flows inside the right atria through the superior or the inferior vena cava. Then the tricuspid valve opens and then it moves into the right ventricle. The semilunar valve open and it moves into the pulmonary artery. From the pulmonary artery, it moves inside the lungs, and through the pulmonary vein, it reaches the left atria. The bicuspid valve opens, moves into the left ventricle. The semilunar valves open and it moves into the aorta. Is the flow clear? Is it not easy? Yes? Clear now? Fine. But it is not that the right part will work and then the left part will work. So both the parts of the heart will work simultaneously. So when blood comes inside the right atria, blood also comes inside the left ventricle. So blood enters the right atria from the superior inferior vena cava. At the same time, blood enters the left atria from the pulmonary artery. Together, the bicuspid and the tricuspid valve will open, and then it moves into the right ventricle and the left ventricle respectively. Then at the same time, the semilunar valves will open and blood from here moves into the pulmonary artery and this moves into the aorta. Both the sides of the heart work simultaneously. Understood? Is it tough? It's pretty easy, right? So I guess the structure of the heart is clear and the flow is also clear. You should never forget this is pretty easy, right? So rather than teaching you from this diagram, which is a bit confusing for all of you, I have taught you in my own diagram. So trust me, this is how I read when I was in class 12. I still remember it. So when you make simplistic figures and study, you will never forget it, okay? So I guess the flow is also clear. So now we move forward. So yeah, whatever I told you, that is written in the form of sentences. So let's read the sentences once. So the heart is mesodermally redived, so it is situated in the thoracic cavity in between the two lungs, but it is slightly tilted towards the left. So protected by the double-walled membranous bag, so that is the pericardium, it encloses the pericardial fluid. So it has 4 chambers. The 2 upper chambers are relatively small, that is the atria, and the 2 lower chambers are relatively big, that is the ventricles. So there's a thin wall between the 2 atria, that is the interatrial septum, and between the atria and the ventricle, you have the atrioventricular septum, and between 2 ventricles, we have the interventricular septum, right? So now, then we spoke about the valve. Now, so with this, the first half of the topic is done for today. So I just have one more bit, that is one last slide for today. So what are nodal tissues? So when we talk about heart, you would have heard the term SA node, AV node. So what are they? What are they used for? Now that we have spoken about the structure of the heart and the flow of blood inside the heart and out of the heart, now how does the heart function? For example, you know, in the motor takes water from one end and puts it on the other end. But how is the motor functioning? With the help of electricity, right? So I turn on the motor. Yes, the motor has the ability to pump water from one end and pull it out on the other end. So now it runs on the basis of electricity. So now how is my heart going to function? So just like that, how blood will flow in and flow out, there has to be some energy, there has to be some force, right? So that is the action potential. So who generates the action potential? So we have a patch of tissue in your heart. So we have a series of patches of tissue, and that is called as the nodal tissue. So once they generate action potential, so that is when the heart functions properly. So without which it cannot just open and close. So even if a door has to open and close, it has to be automatic, right? There is a difference compared to the normal door and an automatic door. So much in the same way, for a heart to function, we need what is called as the nodal tissue. So let's talk about what is a nodal tissue now. So let me redraw the diagram once again. So let me redraw the diagram once again. So we have the right atria, right ventricle. So left atria and the left ventricle, right? So towards the corner, left corner of the right atria, there is a small patch of nodal tissue. So node is a small patch, okay? So that is called as a node. So I have a patch-like tissue that is called as a nodal tissue. So this tissue that is present towards the upper left corner of the right atria, so that is called as the SA node. So if you can see, it is exactly here. So this is the right ventricle. Right atria, right ventricle, left atria, left ventricle, right? So here we have the SA node. So this is the SA node. So SA stands for sinoatrial node. Fine. And then you have the AV node. So exactly at this junction, between the interatrial septum and the atrioventricular septum, there is another patch of tissue. So that is known as the AV node. So it is known as the atrioventricular node. Okay. So now in this wall, so you have what is known as the bundle of His. So exactly in this region, you have what is called the bundle of His. So what is the third one? Bundle of His. It is a bunch of fibers, right? And throughout the right ventricle and the left ventricle, I have another patch of tissue that is known as Purkinje fibers. So what is the fourth one? So that is the Purkinje fibers. Fine. So I have the SA node here, AV node. Then in this line, I have the bundle of His. And then in the ventricles, I have Purkinje fibers. So all of these four nodal tissues, they can generate action potential. They can generate action potential. But the maximum action potential is generated by the SA node. So since this generates maximum action potential, this is known as the natural pacemaker of the heart. Because this can initiate, so all of this can automatically generate action potential, but the maximum action potential is generated by the SA node. Okay. That is why it is called as a natural pacemaker. So this will initiate an action potential, pass it to the AV node. So from the AV node, it passes through the bundle of His. Then from the bundle of His, it moves into the respective Purkinje fibers. So that is how this works. Clear? So this is called as a nodal tissue. So in a minute, your heart beats 72 times. Fine. So this is because of the action potential that is generated. So when they say they have an artificial pacemaker, which means the SA node is not working, so they replace it so that there is another alternate which can generate maximum action potential. So with this, we are done with the structure of the heart. Talking about the flow of heart and what are nodal tissues. So in the next class, that is on Monday, we are going to talk about cardiac cycle. So you would have heard about the term what is systole and diastole. So that's what we're going to talk about Monday. So Monday we'll talk about cardiac cycle followed by ECG, so that this chapter is done. Okay. So I guess the structure of heart, I have made it clear. So any doubts that you guys have? Any doubts? Hi Dagni, yes, after a very long time you are here. A very, very happy good evening. Yes, I'm doing good. How are you? I hope you're doing great. So we're almost done with the lecture. So I guess you should watch the recorded version. So we spoke about the structure of the human heart. You made the confusing heart to the easiest. Okay, so it's my pleasure. Thank you too. Yes, so Bhavya is also fine. So she has also understood. Fine. So please go through this PPT. So I guess they have uploaded the previous one. So please go through the PPT and remember this diagram. So this was always gonna help you because this has helped me all through the years. I never forgot the structure of the heart. So this is one easy way of remembering. So please stick to this. Otherwise, if you know it, it's fine. But this is one easy way of remembering. So I guess you remember the summary. So we started talking about what is lymph today. So lymph is the tissue fluid. So it is present inside the lymphatic system. It also plays an important role in the defense system. So then we spoke about what is open and closed circulatory system. So we spoke about the examples of each of them. Then we spoke about the structure of the human heart. So first we spoke about the structure, the membrane, the walls, etc. Then we spoke about the flow of the human heart. So the flow of blood inside. So in which direction it flows. Then we spoke about the nodal tissues. Right? So this is what we spoke about fully. So I guess this is the summary. So yes, ma'am, good. I will watch the recorded session. All right. Okay, Dharni. So Bhavya says no, they have not uploaded. So let me open the app and check. If not, I will tell them right now, and end to end they will upload it. Okay. So with this, the class for today is done. So you can have a beautiful weekend ahead. So please do enjoy your Sunday as well as do some homework as well. So solve some questions. If you have any doubt in certain questions, so bring it to my knowledge. I will ask them to put the link for Telegram group so that we can have one-on-one interaction as well. So with this, I come to the end of today's session. So thank you so much for joining me on the Saturday evening. So I hope all of you have a most amazing weekend ahead. So hoping to see you on Monday. So until then, this is me wishing you more and most. So until then, take care and bye.